Hello and welcome everyone in another episode of Researcher Celebrity. Today we have Dr. Sabyasachi Pradhan with us as our Researcher Celebrity. A brief about Dr. Pradhan. He has done his bachelor's from University of Calcutta and master's in biotechnology from MIT University Noida, following which research became his passion and he did PhD from Jawaharlal Nehru University, JNU, New Delhi. Then he joined as a research scientist the Tata Institute for Genetics and Society and visited UCSD, University of California, San Diego as a visiting scholar during that same program. Presently, he is a research associate at University of Glasgow. With this short brief about him, I would like to call Dr. Pradhan at the platform and welcome you.
Thank you, Raja. Thank you so much for having me. Great to be here and great to be talking with you after a long time. Thank you.
Yes. So, Sabir, how we normally start the conversation is like how and when you decided that you wanted to be a researcher.
Very frankly, I probably never decided that I want to be a researcher, but I wanted to be in a academic field mostly. That's always has been my passion to stay in the academic field, and probably that's the reason I still am working in a university in a hope that someday I might land up an academic job with obviously the research option open. But my parents are doctors, and so was my grandfather, who passed away a couple of years back. And I have been growing up around diseases most of my life. I think almost all of my life, I am seeing them treating patients, and mostly patients with tuberculosis. In Calcutta at that time, there were a lot of patients with tuberculosis, and it was one of the diseases which really intrigued me. And I wanted to do I wanted to do my PhD in tuberculosis, which unfortunately I couldn't. So I landed up working with malaria. But to be very frank, I actually wanted to be a doctor myself. I tried the exams and unfortunately didn't work out. And then I fell in love with microbiology. And I just couldn't stop myself looking into the microscope and looking at the tiny organisms which are responsible for causing diseases, among other things. And so I thought, okay, if I can't be a doctor, like a medical professional, I can definitely contribute to the field of diseases from, the other side. Let me do research and try to, find out some interventions which might help to tackle the diseases which I am seeing my parents and my grandfather tackle their whole life as a medical physicians. So when I was struggling to get a PhD option in India, way back at that time, things were much simpler. I was one of the person who, from my batch, probably the only person from my batch who cracked the NET exam in the first chance. And I am absolutely certain that was just by luck, because we had really very good students back then in my batch. So when I was still struggling to get a PhD option in India. I was roaming around in different universities, sitting in the exams, in the interviews. So my mentor, Professor Unni Kumar, uh, unfortunately, he has also passed away. I really hoped he could see me right now, trying to do some research. He really was always backing me up to go into this route. And he actually suggested me to go and work with malaria. At that time, I remember him saying that, Bill and Melinda Gates are paying a lot of money to do research in malaria. Why don't you just go and visit Dr. Tridivesh Adak in National Institute of Malaria Research? I went to Dwarka from Delhi. It felt like I was moving from a city to a very barren land. There was probably this only one building in the whole of Dwarka Sector 9, which was the building of National Institute of Malaria Research. I met him, what I think on the 7th or 6th of September 2009, and I joined NIMR on 7th of September. I was really intrigued by the work he was doing. And basically, I thought that I really could contribute, if not to tuberculosis, but to malaria, is which was and is still one of the like largest diseases which is killing a lot of people, at least not in India, but in Africa, in Southeast Asia, in other countries, there's still a lot of mortalities happening in malaria, especially with infant and pregnant women.
Great. So just in one meeting, you make up your mind that you want to do a PhD in that lab?
Well, Alexa was convincing, uh, and he asked me, "Would you like to do field research?" And I said, "Yes, of course." I although I really like looking under a microscope, which I am still doing, I would be doing tomorrow also. So, and he said, "Would you like to do field research?" And I have always had a keen interest in doing field research. You know it very well. We have collaborated in the past doing field research. So that was one of the selling point for me. And he showed me around National Institute of Malaria Research. It was a brand new building that time, lots of new instrument. And for a fresher was just coming from doing his master's and not getting a lot of chances, I just couldn't say no. And I like malaria. I studied about it during my graduations and post-graduation. It was a very difficult disease. It was a disease uh, caused by a lot of different like pathogen, but there were multiple pathogens that could infect humans. It could infect birds. It could infect rodents. So I thought it might be a good option to study, and also it would give me opportunity to learn a little bit of the about the mosquitoes also, which apart from having been like vectors to parasitic diseases, also is a vector to viral diseases like Zika, chikungunya, which and dengue. So, and those diseases can also be tackled. So I thought if I can learn about both the vectors and parasites, I might be able to focus my, research interest into solving the problem for the poor people mostly, because those are the people who really needs for like we need to have very easy and very cheap interventions developed for them so that they can afford them in underdeveloped countries where people do not have the infrastructure. If we can find a way to make like good products available to them much cheaper, which could have longer shelf life and could be used by semi-trained professionals, that would be a really nice boost to everybody.
That's great. In fact, that being said, malaria is the top neglected disease which people are studying and working for. So this needs a very passionate and compassionate researcher to be, because the way you're talking about diseases, the way you approach them coming from your father, from your grandfather, because you were in that environment. Because people still, most of the people want to do all the fancy research with top course, either it is the cancer or others. But when researchers like you come to the field with this pure intention of helping people, that's what make a significant change in the society as well as the research community. So Sabya, if you can share that, how was the journey of your PhD?
My PhD journey was a roller coaster. It was, well, how do I say? It's probably one of the longest I have studied in one place. It took almost 7 years, and I had to go from one institute to the other. It wasn't very easy uh, because things back then were not very streamlined in India. Or unfortunately, I wasn't in an institute where things were streamlined. Fortunately, things are very well streamlined right now for all the newcomers who are joining their PhD in different universities and different institutes throughout India. things are way more laid out for them, and it's just you concentrate on a research and you can get it done by time. In my While I was doing, things were not easy, and also because of the reason that I got dragged into this bet with my professor, Dr. Adak, at that time, that he really wanted me to establish the vector parasite interaction, which was like the P. falciparum, the Plasmodium falciparum, and Anopheles crucifixus or Anopheles stephensii feeding model. So this is a model where you culture your parasites, the malaria parasites, in lab, and then you can artificially feed them to the mosquitoes, and where you can study their sexual cycles. The sexual stage happens of the parasites happen inside the mosquito. And this whole infrastructure is something which is not present in all the malaria labs. Very few labs in the whole world has this kind of infrastructure. The lab where I am has, I know lab at John Hopkins has, and there are few other places in the lab in the world where they have really good infrastructure where you can study the vector parasite interaction. So that took me a long time to figure out how to do it. And that was not very easy. It was very difficult to set up this infrastructure back in India. Fortunately, I got the opportunity to come and visit my current supervisor back 10 years back in Glasgow. So she showed me how the things are done, and I went home. I went back to India and tried to replicate. Unfortunately, I wasn't successful, but at least I got to learn a lot of the things which I have, like what are the things I need to know about the parasites and how to culture them and how to work with them from her. And it was very important in order to complete a big part of my PhD. And since I wasn't able to, replicate the sexual cycle stages with the Anopheles crucifixus or like Stephensii with falciparum during my PhD, I had to switch my focus from the human malaria parasite to the rodent malaria parasites. So they are genetically not similar, but a lot of the falciparum behavior can be extrapolated from studies done with Plasmodium berghei, which is the rodent malaria parasite. So we collaborated with Dr. Agam Singh at National Institute of Immunology, and we started the gene PCNA2, and we wanted to see how that gene was involved in the parasite DNA replication. So that took some time, but I think that was the biggest good news that could come in my PhD. Working in his lab helped me a lot to learn about the new techniques like transfections, a lot of other stuff, how you do how you do rodent malaria parasite cultures and handling mice, and a lot like mosquito maintenance, etc. Dissections, sporozoid dissections. So those are really helpful working with him, and that led to me finally culminating all my 7 years worth of work into my research thesis. So yeah, it's not been a very easy journey. It was always working apart in National Institute of Malaria Research, doing a bit of work with Professor Sumandar. Obviously, I should mention him. He is actually my supervisor in my PhD, and actually Suman sir really encouraged me to go and work with the rodent malaria parasite, seeing that I was struggling so much trying to establish the falciparum model in the lab. So I did go a lot far than I intended to go with the falciparum model, but somehow it wasn't working to the extent we needed to do our experiments. We were also having some problem with falciparum transfections. So one thing led to another, and Professor Dhar really encouraged me to go and work with Dr. Agam Singh in National Institute of Immunology to make sure that I could replicate the work I was planning to do with Plasmodium falciparum, and we could do that work with Plasmodium burkei. So one thing led to another, and it was, I think, working with three different PIs who has three different, very unique ways of handling students and tackling problems. Although it took a long time, but I really learned a lot from each of them separately. And I think I don't have any regrets for it. Only regret I could have done a little more from the publication point of view, but yeah. But otherwise, I think I am happy. It took a long time, but at the end, if you keep on going, keep on pushing it, you will succeed. So and I think if I could succeed, anybody can do a PhD and succeed. Just have to be patient and just have to be focused, and you will succeed, definitely.
Nice. So, Sabya, the success and the struggle of PhD, you did it with three different guides, which is extraordinary because not many people can do that, because people are either bored or end up in arguments when they are working with more than one PI, but you did it with three of them. What are the suggestions or recommendations which you can give the researchers or the young researchers who are just studying their PhD, that there are what they should do to avoid the struggles, or to minimize them at least?
I believe nowadays things are much more streamlined than the time I am from. People, PIs nowadays, even like supervisors and the students, especially the students, are much more focused at what they want to do with their study. And they have a clear-cut plan laid out from the beginning. And that has caused, I do not believe that nowadays you won't be needing such long times to complete your PhD. It was just that probably I was too adamant that I could or like I should be doing this only, that maybe did not work to my advantage. If I had switched to the stuff uh, or to some other things when things were not working with setting up that in vitro falciparum culture, it might have been a different story. But I didn't do it. I thought that I could do it. And because that was kind of the thing that I always wanted to take up challenge and solve thing. And until I could not solve that, I did not want to take rest. So that was my, strength and weakness at the same time, that if I would have selected something a little bit easier, maybe I could have done it. But yet I did not want to select something easier. Now, right now, I think any student starting or in the middle of the PhD, there will always be a little bit of difference with your supervisor. And I think that is common and that should happen. That will give you an, idea of how you should deal with your students when the time will come. So I have taken, like working with three different supervisors, I have seen how each of them have different problem solving attitudes. And I have taken tips from all of them, which actually helps me to solve any problems which I'm having with my current supervisor, or I am having some problem with my colleagues or maybe juniors. I really have learned a lot of these things by talking with them and learning from them, and which has really helped me. So there will be little bit of you know tips with any PhD student and supervisor. I don't believe that any student and a supervisor will always have a great relationship, because a student might think something is possible and the supervisor might think that is not possible. And as a student, you are quite young. And believe me when I say that I was a student. So I know what you are thinking. And when I was a student, might have approached someone and said, hey, can we do this? Can we make this like DNA library prep or something very fancy thing I might have read? And supervisor might have turned me down. And I did not like it. But now I understand that a supervisor just doesn't turn you down just by itself. When you're young, when you are just out of your master's, you read a lot, you have this lots of ideas of what you want to do. Nowadays, there's so many different techniques, so fancy techniques. You can do so much stuff with so little starting material for any, I think, any biological researchers, maybe any other physical or chemical researchers. So you are like filled with ideas, but the moment you start doing the work, it you will have a reality check. And I wouldn't wish that anybody have a reality check like I had when they have given a lot of time doing a process that is not very easy to do. So I still should think that you should listen, everyone should listen to the supervisors and take note from them, and then try to come to a middle path, a middle ground of understanding where both of your wishes come true. I know it's very easy to say because I have moved away from that position. And while I supervise students now, I can really understand where the student is coming from. And they might not like what I say, but they also have to understand that I say things from experience.
Yes.
So all the supervisors say things from experience. And you also have to put yourself in the shoes of the supervisor and understand that it's not very easy to do research, and especially when research costs so much of money. So you might think that my supervisor is not giving me this, or my supervisor is not wanting me to do this, because that might cost a lot of money, and you probably are not the only student he or she is supervising.
Mhm.
So they have to look out for all the students equally. That is their job. And they may or may not have the resources to do a fancy experiments. But yet again, not all fancy experiments will give you results which might help you with anything. Simpler experiments can get you much better results than a fancy experiments with results you may or may not able to analyze properly. So being a supervisor is very tough. I can understand now. While I was student, I probably had the same thing which you might be having as a student, that, oh, my supervisor is saying this. It's not possible. It never happened. But trust me, they are coming from experience. Whatever they're saying is experience. So you should listen to them. And I think you should, talk more frequently with them and try to get or try to understand from what point they are trying to tell you. So if they're trying to tell you that this experiment is not suitable, I think you should have a dialogue, a proper scientific dialogue with rationale. And I believe that if there is rationale in your request, no supervisor will say no to it. They the only reason they might say no, because they might feel that you are going on the not on the right path, and maybe you might not succeed and you might be disheartened with that, and nobody wants to see that. I don't think any supervisor wants to see their student disheartened.
Yes.
So they are there to encourage. Even in their no, there is encouragement. So do talk with your supervisors. Don't just be sad or angry with them. So coming from me, you should know.
It's a bit, this is something which I think I should just take this time to pause and tell all the audience and viewers here. Sabya essentially what just said is you should be more sympathetic and more understanding to your supervisor, which can resolve your conflict with your supervisor and can keep you both at the same page, because that is very important. Sabya mentioned about the experience your supervisor had, and that's the reason why they are your supervisor, because you do need guidance from them, or you can call them mentor, because they know some of the possibilities which can be helpful for your PhD, and some of the things which can prepare you for the future but are not correct at that point of time. So, Sabya, when you finished your PhD, what was the first thing that you were thinking when you were submitting the thesis?
First thing when I was submitting the thesis, I think it was finally it's done. I think that's the first thing I was thinking, like finally it's done. I can have the, call myself Dr. Sabyasachi Pradhan. Not a,
Mhm.
doctor as every Indian wants themselves, not a physician, but at least a doctor. But I think I was a bit relieved when I submitted my thesis. So it was long awaited. My parents were relieved. I think my mom was relieved much more than anybody else. So yeah, I think I was a bit relieved when I submitted my thesis, and a bit sad too, because I know that after you submit your thesis, then a different phase of life will start. You will no longer be student, so no more student discounts in a lot of stuff. Not like, discounts, but you do get a lot of discounts in life when you're a student. So a new phase of your life starts after you submit your thesis, which probably is the hardest because you don't get to have your batchmates around you a lot, like at all. They probably will end up at different places in the whole world. Like most of my batchmates are no more, hanging around with them at work, after work. It becomes a totally different lifestyle after you've completed your PhD. So I think as long as anybody is a student, I think that's the best time. So it was bittersweet. I was happy as well as sad. Then I won't be coming into work or like coming into the lab every day, seeing all the people, the support staff, the our admin staff. So now I see a support staff and admin staff, but in a different way, not as a
Mhm.
student, but as a staff, as staff myself, or as a research associate or a postdoc myself. So it's different. But yeah, it was a bittersweet moment when I submitted my thesis.
So, frankly speaking, even for me, I call the best time of my life were those years when I was doing the PhD. And when you did your PhD, you moved to Tata Institute for Genetics and Society. How was the experience from moving from Delhi to San Diego?
Nice, actually. Uh, the weather was fantastic.
Mm-hmm.
And like probably like I would say I probably that's why all retirement homes are in San Diego. If you the weather's fantastic, if you're a person who likes sea like I do, like water. I'm a fan of water, not as much as mountains. So I like water. So I just saw when I googled San Diego and saw in Google Map where it is, and I saw the Pacific Ocean is just a stone's throw. Literally, if you are strong enough, you can throw a stone and it will land in the Pacific. So, and I said, yes, enough. That's the place I have to go. There is no other options. Just have to crack this interview, and that's the place I have to go. So San Diego was very nice from the weather wise. Uh, Laboratory wise, it was again, I don't know, it's just bad luck follows me everywhere. I was deputized to work with Professor Vinets, Joseph Vinets, but unfortunately, he moved to Yale from San Diego, and I had to instead of getting to learn a lot of things which I assumed I would be doing from him. I was thrusted into setting up the whole infrastructure to test genetically modified mosquitoes as well as to test different interventions like monoclonal antibodies and nanobodies against malaria. It was a very, I wouldn't say difficult, but very challenging to do, to start anything from scratch, especially in a country where you have never worked before.
Mm-hmm.
You do not know the regulations, rules and regulations, which are very, very much different. And way difficult than working in India. India is catching up very quickly, that I must say. But at that time, setting up something from scratch was very difficult. I had help from few people. I would like to mention Dr. Kailash Patra. He was there to help us, not me, but help all of us to help this set up the insect tree and set up the parasite culture system. It was great working with him for the brief moment of time. And he was also a mentor to me at that time in San Diego. The work culture there was different. And since I was not a student anymore, it was more result driven and focused work. But once we got our things going, I think that was the time I really started to enjoy working. I think, yeah, we got to test a few of the antibodies, and we got to test genetically modified mosquito with our setup. But finally, when we are ready, test more, or do much more detailed testing about testing the nanobodies and other stuff. Something happened, it was March of 2020, and I think the whole world shut down. So basically, we built the platform, but I was very unlucky to do any testing with it. So that just leaves a bitter taste in my mouth, that it took a lot of time, like mostly 2 years, to set up something, and I couldn't get anything done, or I couldn't do anything with it. But I hope the people at TIGS did some work with it, otherwise it would have gone in vain. After coming back to Bangalore, I still worked with TIGS for a year before I got this opportunity. But that time also, it was still the COVID wave was continuing in India. So it was a very difficult time. And since my expertise has always been with wet lab work, and almost every day I have to go to work, including Saturday, Sunday, to do my cultures and take care of mosquitoes. It was very difficult to have something concrete in that period, because we weren't allowed to go to work. Like, as far as I remember, I think it was once, like, you work for a week and then you take break and the other person works. So there was a lot of disruption. And finally, I saw that things weren't going any further. And the institute had other plans. I think they wanted to move away from the malaria project to something more tangible at that time. And since it was not resonating well with my expertise, I thought that it might be wise to go and work in a field, or go and work with a person with whom I can, who can help me learn a little bit more about the work I have already been doing, to work with Plasmodium once again properly. Fortunately, I got this job opportunity in Glasgow, University of Glasgow, with Professor Lisa Ranford Cartwright, in whose lab I already learned how to do parasite culture. So it's nice coming back here after like 7, 8 years. And I think I have to thank her a lot, because that small stint working here, a month and a half in her lab while I was doing my PhD in India, really helped me open my eyes to how things operated in the world stage. So how people do their culture, how, how do you do your science, how you should behave yourself in lab, which us Indians most often don't do. But so it's not only science that I learned in a lab. I learned a lot of other things other than science, how to carry yourself as a scientist, or how to carry yourself as a PhD student, and how to maintain your notebook, how to maintain where you are keeping your frozen stocks. Oh my God, she really has, like, if I hadn't seen those things, I
Mm-hmm.
might not be sitting here talking to you. And that I can say for certain. So there has been a lot of event in my life, and I think coming to her lab, that was probably the most crucial event I can say. So that changed me a lot when I came to her lab, and probably I have to thank my wife for that. She somehow convinced Dr. Adak that I deserved an opportunity to go and visit her. And Professor Jane Carlton was there in the building, and Dr. Adak convinced her to give me an opportunity. Well, she gave me an opportunity to come visit Glasgow. And only thing I regret that I couldn't go back and replicate the work. I really tried, but things just didn't work there. Things were different. The parasite strains were different. The mosquito was different. I couldn't figure it out, but I did figure it out. So we did set up the whole infrastructure back in San Diego, obviously with a different mosquito strain and a different parasite strain from another lab. Things were different, but I knew how to do the things. It's just it didn't work out at that time in India. Probably if I go back now, I absolutely have better chances to set that up. And I'm hoping I would get that opportunity to go back when I'm or maybe or somewhere where I can set that up infrastructure and do some research after I'm done here. So
Sweet.
yeah.
So, Sabya, you just briefly mentioned about your wife also, and I know her. So, how does it feel to be uh, like when your spouse is also a researcher? How easy or tough it is?
Well, it's both easy and tough. So you can't fool your spouse when you say, "Oh, I am doing a fancy experiment," right? She will know that PCR doesn't take that long, or
Mm-hmm.
so she will know from that way. But when you're struggling with PCR, you can come home and ask, "Hey, I'm struggling, help me out," and
Mm-hmm.
she will. So that's the benefit of having a researcher in the same field, or at least a person with whom you can talk with your problems in the office, and you can actually get some constructive criticism, or like not criticism, constructive feedback on how to tackle problems. And it goes both way. I'm not saying that people in different field can give you, like, cannot give you any advice or not, but something like, instead of going to a friend or someone, you always have your spouse to help you
Mm-hmm.
out in situations if you are stuck. And it also helps you, and it's just like you have an extra pair of eyes looking at your work from the outside, which
Mm-hmm.
always helps, which I believe always
Absolutely.
helps, because it's a neutral pair of eye, which is some extent biased to you, but obviously looks at more as a neutral than
Yeah.
totally, like, because if I look at stuff, I might be more biased to my experiment than my wife is. So yeah, uh it's not also there are, the thing comes that if your spouse and you are on the same field, there might be an ego thing coming, of
Mm-hmm.
which is quite possible, which absolutely is not a case with us, not at
Mm-hmm.
least with science, because she is way too intelligent than me to have any ego. So, yeah, so I think it's just as a person, I think if you can keep science at work when you come back home and maybe only discuss it if it is absolutely necessary, I think
Mm-hmm.
it's all right. It's
Yeah.
perfectly fine to two-person. And I think when you know someone for a long, long time, things just works much better instead of if you knew someone, for a short time in the same field compared to, like, I knew her since what, I forgot, it's so long. I knew her since 2009. So, but we got married quite late, but I knew her since 2009. And you wouldn't believe me, my first day. I think my first day in an MR, she asked me to, well, it's quite embarrassing, but anyways, she asked me to, reconstitute the wash buffer for a Kyogen DNA extraction kit. So I went to the neighboring lab to get ethanol, add wash ethanol to the wash buffer. And it was like a 250 reagent kit, 250 reactions kit. So it was a big thing. And I just grabbed a bottle of ethanol from the fridge. And as I was young and naive, I did not read that it's not ethanol in the bottle, they kept something else. So basically, I added fixative, which was used probably for fixing embryo stuff. So I added like a whole bottle of fixative. And then I realized I needed more. So then I went to her to ask, can you give me more ethanol? And she said, where did you find the rest of the ethanol? I said, it was in the fridge. Then she said, ethanol should not be in the fridge. What did you add? So again, people, when you're adding some stuff, read the label 10 times. And if people are reusing bottles, please remove the label and write what you are having it properly so that person like me does not make a mistake. So yeah, she scolded me a lot at that day. Well, it's India, your seniors are allowed to scold you. But that was something. So yeah, it started with something like that, and then we are here today.
Mm-hmm.
Blessed with a baby boy last year. So things fell into place. So it was my mistake that I added fixative to a 250 reaction Kaizen kit. And I must say, Shalab really came through. You know Shalab, right? So he replaced, I think, the bottle and gave us a new wash bottle. I don't know how he did it, but he did it. So
No, this
yeah.
is very important for all the researchers, especially the young ones who are just joining the lab, to read the labels, and for the people who are working in there to remove or at least rewrite if they have changed something in the bottle. This is
Properly.
very
If you're
important.
reusing a bottle, please
Yeah.
properly do it. So you might not be as lucky as me. You might not
Yeah.
end up, having a life partner. But
True.
anyways, I got lucky.
So,
So
Sabya, at
I
the
can't blame.
yeah, so we are at the edge of the conversation now. What would you suggest for a student who is considering to be, like, whether they want to be a researcher or not? What are few bullet points, or in one sentence, if you can say that, what they should have if they want to be a researcher, or what they don't?
I think they should have patience.
Mm-hmm.
That's probably the most important thing, Rika. So, research means you are research. So you're searching again. Somebody has searched it. If you don't have the patience to search again, then this is not the field for you. Reason I am saying because my work needs a lot of patience. It's just my experiment starts in day zero and it takes me a minimum of 5 weeks to get anywhere near my result. And if I need to modify anything in that experiment or that protocol, it requires another 4 weeks to 5 weeks to see if I have got, like, if that modification has helped or not helped or whatever has happened. So it takes a long time. And I'm not saying not all research is like this, but research by default needs patience. You cannot hurry up something, especially when you are working with a biological organism like I am. I'm working with two at a time. So I think most of the biologists will work with a biological organism. Maybe it's bacteria, fungi, algae, birds, zoologists, birds. I just went to Isle of May, so they work with birds. So you really need to be patient with any organism you're working with. You have to understand that the organism, you have to understand that we need to study the organism, not the other way around.
True.
So we have to modify our schedule according to them. It's not the other way around, right?
Absolutely. That's what
We
how you
grow,
ended up doing on weekends and on
yeah. So
Saturdays, Sundays, because you have to follow their life cycle.
we have to follow their lifestyle. We
True.
grow mice We grow mice in labs every day
Mm-hmm.
and sacrifice them,
Mm-hmm.
learn something about some diseases, right?
Mm-hmm.
And I grow mosquitoes in lab every day and just kill them or to learn or like dissect them, which I'll do tomorrow to
Mm-hmm.
see the results of my experiment.
Absolutely.
So we have to pay that respect to the organism with which we are working with, and we have to be very
Absolutely.
patient with them.
True.
So I think in short, if you need to have patience, and if you're working with any biological organism, you have to have respect for that organism
Absolutely.
because if you don't respect it, it will not respect you back. So that works with any cell line, any microorganisms, any macroorganisms. Like I'm right now part in the zoology department, so I see people working with a lot of organisms I never heard of because we probably don't use them in India. So I see people working with birds, with reptiles, with Japanese squirrels, so many fascinating things.
Mm-hmm.
Siberian, some things I just heard yesterday. So these are all like model organisms. So each of the organisms have their own, you know, have their own life,
True.
and we are just utilizing them to find a common goal or to, learn more about certain aspect which would enhance our life.
Nice.
So we need to be patient and we need to be respectful to those biological organisms.
Great.
So coming from biologist, so I cannot say about physics or chemist, but
Mm-hmm.
obviously in those or mathematics or in those cases, I think patience is the key. You need to have a lot of patience. You shouldn't hurry up. And
Mm-hmm.
right now, you should be very much careful in how you are doing work because your publication matters.
Mm-hmm.
Because once you have published stuff, you need to know that they're all okay. And because
True.
lot of people will rely upon your publication and work around it or work with that information. So you have to make sure that information is correct. So, and I think patience is the key. You should not hurry up with stuff. That doesn't mean you
Absolutely.
should take 8 years to do your PhD like I did, but
True.
at least, be patient and things will work out for you. I think that's the thing. And be respectful to biological organisms every
Absolutely.
day. Yes.
Yes, I think I cannot stress more than as you already did to all the researchers there that patience is the key, and what we learned from Dr. Sabya also in this conversation that you should pay due respect to the system you are working with, the biological systems. And with this, we'll say thank you very much, Sabya, for taking time out and guiding so many researchers around the world, because all of us know that patience is the key, but it comes with the time. And if people like you guide them well in a way, so that's how you get benefited for what you have learned, and they get benefited what they can learn from you. So with
Yeah.
this, I know Sabya, and he's a fantastic person, a great scientist. So if you have any problems with your, if you're working with malaria parasite or the mosquitoes, he's the person. Feel free to write it
No,
to
no,
him.
mosquito is your domain. I'll not step into that.
So feel free to write about the malaria parasite. If you want Empowering Science Foundation to help you to connect with our researcher celebrities, please feel free to write it to us. And with this, we'll say to all the audience and viewers, stay tuned for another episode of Researcher Celebrity. And here on today, we'll say thank you very much, Sabya, and good luck for your future research.
Yes, I also want to thank you very much, Raja, for inviting me and giving me a chance to talk. I hope people, uh, you know, uh, well, a lot of people tune into this researcher celebrity thing. And thank you to you and the Empowering Science Foundation to have me as a guest. And I'll be happy to come back if you would like me to, and if
Absolutely.
people like me to. And yeah, and I wish all the students, if anybody wants to do PhD or however way you are going, all the best in all your future endeavors. And you should remember that, science is for everyone. Anybody who has the patience and stamina can be a scientist. So yeah, just keep calm and carry on.
Great. It was a pleasure having you, Sabya.
It
Thank
was
you.
a pleasure. Likewise. Thank you so much.